Okay, uh, let's start. And today, let's be look into a little bit of gene regulation, mainly post-transcriptional regulation. Some of the examples, and we'll wind up the course today. And today will be the last class. And in the following weeks, 1 or 2 weeks time that we are left with, I'll be sending some of the questions, and then I'll compile all the questions that you have framed during your assignments. And I expect people to look into that. Uh, that will have some, this will have some, if there is any exam sooner, then it will have some relevance. Okay, so please look into the questions and try to understand it. If not the exact question, but similar themes at least, I'll try to uh, get some from these questions that I'm going to send in the coming weeks. Okay, so the regulation of gene expression, the earlier times we have talked about in prokaryotes the lac operon, lactose utilization operon in prokaryotes, correct? That I think it's a very beautiful gene regulation, and that was one of the first examples of gene regulation with very beautifully regulated gene system. And of course, we have talked about the Drosophila system as well, correct? For which is required for the segment-based expression of genes patterns in Drosophila. And mostly that is involved in the regulation at the transcriptional level. Either it changes the DNA profile by epigenetic modification, which have a lot of implications in binding of transcriptional factors, either to inhibit or repress transcription, or something with activators can be attracted by these modifications. And transcriptional factors can also have a lot of that can interact in different ways in different tissues and can give rise to different set of gene expression with few transcriptional factors that we have talked about. And of course, we also talk about histone modifications that may have implications on transcriptional regulation. Today, what we are going to do is we will discuss a little bit about or some examples of post-transcriptional or translational co-transcriptional regulation. Okay, so one of the very good examples of co-translational regulation in prokaryotes is the tryptophan utilization operon that we talked earlier. Also, I think we'll just go through again in a better way. So what we have talked about is that the tryptophan utilization operon or synthesis, the tryptophan synthesizing operon, biosynthetic operon, is highly linked with the translational rate of translation. Okay, so here to transcribe the translation, to transcribe the tryptophan biosynthetic genes, what is required is that it starts transcription with leader peptide. Leader peptide is a small peptide which is encoded by the transcript ahead of the tryptophan biosynthetic genes. Okay, so here what we have to note down is the elements or sequences which are present: 1, 2, 3, and 4. If you look at the level, these are very, very important because 1 and 2 uh, can be involved in the regulation. More importantly, the sequence number 2, which can base pair with 3, and 3 can base pair with 4. But depending on the translation rate, it will decide whether the uh, whether the loops which is going to form, either the 3-4 stem loop structure will be formed, or 2-3 stem loop structure will be formed, solely depends on the rate of translation. So if you look at here in the element 1, when the ribosome, as you know, in prokaryotes like E. coli, translation and transcription are coupled. That means before the or when the transcription is going on, translation can also initiate even if transcription hasn't finished yet or completed yet. So in the tryptophan, the leader peptide which is present in sequence 1 has tryptophan in there. So when there is enough tryptophan inside the cell, or in a high tryptophan condition, what happens is that the ribosome immediately translates and passes through the element 1 and reaches the sequence number 2. What does it mean? When it goes fast, the base pairing between the element 2 and 3 in the transcript is not possible. So as the transcription goes ahead, 3 and 4, the element 3 and 4 can form a stem loop structure. And this stem loop structure is followed by a U-rich sequence. So what happens is this is a signal for transcription termination. Okay, so in a high tryptophan condition, the fast rate of translation doesn't allow the base pairing between the sequence 2 and 3 to form base pairing and form secondary structure, stem loop structure. But it allows formation of stem loop structure containing the elements 3 and sequence elements 3 and 4, thereby leading to the transcription termination. So the transcript or the downstream sequences are not transcribed, and downstream sequences code for the tryptophan biosynthetic genes. So in high tryptophan condition, because of these alternate arrangement, the tryptophan biosynthesis genes are not expressed. But when in low condition, in low tryptophan condition, the element 1 or the tryptophan in the element 1 cannot be translated because there is limitation of tryptophan. So it gives more time for the transcript to form base pairing between element 2 and element 3. So what does it mean? It doesn't allow the formation of these transcription terminator stem loop structure, thereby allowing the transcription of downstream genes which codes for the tryptophan biosynthesis genes. So you can see the rate of translation has directly correlate with the transcription of the downstream gene or expression of tryptophan biosynthesis genes. Next is the arabinose utilization of operon. So here we need to look at is how the arabinose is utilized inside the cell or biosynthesized in the cell. So in this case, what needs to be noted here is that there are two operator regions. The operator regions are sequences wherein accessory repressors, or mostly the repressors or activators, bind and affect the expression of genes. And the inducer here is more of, it acts as an enhancer for the transcription. So, and then promoter, PBAT promoter, for that will be analyzed in the next slide. So what needs to be noted is for aravino's degradation pathway, or to utilize aravino's, you need to look at the operator regions and inducer element, that is ERA1, ERA01, and O2, and PBAT promoter. Why it is important here is that the operator 1 and 2 here, these are the sites wherein it is going to bind by a protein called ERA-C. ERA-C dimer acts both as activator and repressor depending on which operators and which inducer elements are being bound by this ERA-C promoter. And the PBAT promoter, what we are talking is the promoter which allows the utilization or degradation of the arabinose, ARA-B, ARA-A, and ARA-D genes is regulated by the promoter around here. So why it is important here is that this, when ARA-C, okay, binds to the inducer element 1 and operator O2, this one O2 and I1, it becomes repressor for these arabinose protein gene. Okay, so when the ARA-C levels, so it allows proper regulation. If there is no arabinose, what will happen? The PBAT from ARA-C binds to O2 and I1 and doesn't allow the utilization or expression of ARA-BAD genes, which are utilized for which are required for the utilization of arabinose by the cell. But when there is arabinose, what happens is that ARA-C binds to the inducer elements, okay, inducer elements I1 and I2 instead of O2 and I1, okay, but when ARA-C is there, it binds to inducer element 1 and 2 and allows the expression of ARA-B AD genes, okay, which allows the utilization of arabinose. So this is kind of lactose. Arabinose is again a carbohydrate moiety, so that can be utilized as a just like lactose operon, it can be utilized by the cell instead of glucose. But when there is a glucose, what happens is that the expression is regulated by CAMP level at the CRP element. Binding of the regulator at CRP site doesn't allow expression of PBAT. So unlike the lactose utilization operant, wherein we saw that the repressor is only has a monofunction, it just acts as a repressor. But in the case of arabinose regulation, utilization regulation, ARA-C act both as activator and repressor by binding to O2 and O1 in one condition when there is no arabinose. And when there is arabinose, it binds to I1 and I2 and allows the expression of allows the utilization of arabinose. Okay, now coming back to post.
Excuse me, sir.
Yeah.
In the previous slide,
Yeah.
Sorry, the previous slide. So there's one. No, sir.
This one or this one?
The next one. This one. There is a promoter at O1 site also, sir. So what is that promoter for?
It's kind of the O1 also. It's for the CAMP binding that regulation. It helps in that. regulation.
Okay.
Yeah. So it's
Okay.
kind of auto regulation. It enhances the activation level. Yeah.
Okay.
So then, if we talk about post-transcriptional regulation, before going into that, uh, as we all know, the regulation of gene expression can happen at the transcriptional level by changing the chromatin organization or by changing the epigenetic modification on the DNA or histones that changes the transcription rate. In eukaryotes, we do have RNA processing. Even in prokaryotes also, it is there. But for the protein coding mRNAs, you know that eukaryotes have the mRNAs has to be a matured one after splicing. Splicing can be modulated, and then after splicing, it has to be exported out of the cytoplasm, correct? Exported out to cytoplasm. That again, uh, it can be a source of regulation of gene expression at a level. And third one, after it's being exported out, where does it localize? It can be localized in PWE interacting proteins that we PWE RNA interacting proteins. All those things can have a profound effect that we know of. So all these things, there are different levels of regulation, basically at the transcription, at the chromatin level, and then you have the transcriptional rate, then processing, then exporting out of the nucleus, then after that recognized by the translation. So when we talk about post-transcriptional regulation, it's more of the splicing can be regulated. We talk about alternate splicing, which can change depending on the tissue and depending on the condition, depending on the stress level that the cell comes across. And then once it is exported out, again, if there is that we talk about the ribosomal RNA maturation, depends on the nutrient status. All those things are part of regulation. But here, what we are going to do more is more of translational regulation, some of the basic concepts with some examples. Uh, so one example is we talked about RNA modification. RNA modification can have a lot of implications. In this slide, what we're talking about is that there are many methyl transferases for the RNA as well, and, not only the DNA. So M6 methyl 6-methyl adenosine can have, for example, here the gene here HN RNPC complex binding. What it does is that if there is a modification, this complex RNA binding protein complex can bind more efficiently when there is a modification. So this affects in the alternate splicing of many genes, something like SRSF3. So it can regulate, the regulation of splicing can happen through uh, the post-transcriptional modification of the transcript itself. Okay, so many times this kind of alterations is co-transcriptional, because we know that splicing happens even before the completion of transcription. So at the transcription level, it can change the splicing pattern. It can be alternate splicing, or slows down the rate of splicing. This can happen. And another second thing is these RNA modifications can help, for example, the YTHD1, F1 binding can enhance the binding of translational components, and this can help in either enhance translation or enhance degradation of the mRNA. We are not going into too much detail here, but it can change the pattern in many ways. So what is important here is that it can enhance the lifetime of mRNA by modification sometimes, or even it can go the other way also. It can reduces the lifetime. It can enhances the recognition by the ribosomal complexes to be translated more efficiently. This can happen. So there are RNA modifications which can change the stability or the efficiency of translation or lifetime of the RNA, or the half-life of the RNA can be changed with these RNA modifications. So one of the example here we are talking about is one of the very important protein, that is SOX2. SOX2 again, during the when it has to enter into differentiation state, when you want the stem cells to get differentiated into certain cell line, cell lineage, what it needs to do is it has to change the expression of those stem cell markers, correct? Like SOX2 has to go down. So what it has been observed is that the methylation of SOX2 or some of the stem cell marker gene mRNAs, the methylation or the post-transcriptional modification of mRNA enhances the degradation, thereby allowing the cells, the stem cells, to become committed committed cells, or it's called a prime state. Okay, so uh, modification plays a big role in the maintaining or maintaining the stemness or exit of out of the stem cell behavior. Okay, then next is another very important thing is, which has been discussed little bit, is the internal ribosome entry site, IRES. Okay, IRES, as the name indicates, internal ribosome entry site. So what happens is that many a times in many, especially the viral RNAs, of course, in the human genes also there are is that many of the RNA or plus Can you hear now, guys? Hello.
Yes,
Yes,
sir.
sir, we can
Yeah.
hear you.
Yeah.
Okay.
Yes, sir, we can
Where
hear
were
you.
we? Which slide do you remember? This one. This one was there. Okay. Is this where we? Is this the slide we were talking? Can you tell anyone?
Sir, before the previous one, I think. Could you book over?
This one?
No.
This one?
No.
This one?
Yes.
Okay. Okay. Then let's go back further. So what I was talking was this one during the regulation for gene expression. One thing many viruses utilizes is the internal ribosome entry site IRES. Because of why they like to utilize is that the cellular mRNA translation heavily depend on M7G cap. So what they do is the strategy by the virus is that you doesn't allow the M7G cap base translation and allows the loading of the ribosomes directly by recognizing these secondary structures. And it has been shown that it is more like it mimics structures which recruits ribosomal like for initiation competent complex, that 43S complex that we talked during the translation in eukaryotes, correct? So it doesn't require M7G cap to initiate from internal ribosome entry sites. So because of that, a lot of viruses utilizes these structure for its translation. And okay, and next one what we were talking was this one. Like we talked earlier during the gene regulation for GCN4, many a times the translation is regulated by the presence of multiple number of short operating frames. The translation which can be uh, terminated at certain point, and this changes the uh, probability of the translating ribosome to reach the proper canonical ORF of GCN4. So genes can be regulated at different stages. For example, the phosphorylation of EIF2 alpha can change the number of ribosomes which can be loaded in this GCN4 mRNA. So it can be regulated at this level again. Then next, what I was talking was that at the translation level, you can mask the initiation factor assembly. As we all know that M7G cap is recognized by the cap binding protein, which interacts with initiation factors EIF4E and 4G, and as well as 4A. So that circularizes the mRNA and it's ready for translation. But many times something like here, example here, CPEV factor interacts with masking, and this masking can interact with EIF4E and doesn't allow EIF4G to form the initiation complex, thereby inhibiting the translation from this particular ORF. But when this cell progresses to uh, a certain stage of cell cycle. In this case, aurora kinase is, I think, is a cyclin dependent kinase, and that phosphorylates the CPEB and doesn't allow the interaction with EIF4E with masking. So this phosphorylation releases the EIF4 to interact with EIF4G, and now the mRNA is circularized and it's ready for translation. Okay, so that's one kind of regulation by masking the formation of initiation complex. And this one we talked earlier a little bit about sexless gene, but what it does is that the sexless gene, SXL gene product, the protein, doesn't allow the translation of an mRNA, which is specific for male cells. What it does is it does by two mechanisms. One is it interacts at the 3' UTR as well as 5' UTR and doesn't allow the binding of 43S, that is ready to be initiated ribosome complex, doesn't allow ribosome to translate. One, that is one way to uh, regulate. Another way is the binding of SXL allows the inclusion of an exon, which leads to the uh, uh, which leads to the less stability of mRNA. Okay, and because of the presence of SXL in female cells, the male specific genes are not translated in the female cells. And because of the absence of XSL, in the male cells, the male specific genes are translated. Now, the translation, whether there are many quality control mechanisms present inside the cell. One is NMD, that is nonsense mediated degradation. Second is no stop decay. And third one is no go decay. Okay, the difference between these three is that one, in this case, in the nonsense mediated decay, there is an extra stop codon in the middle of mRNA ahead of the natural stop codon coding for the protein. In the case of no stop decay, what is happening here is that the mRNA which is being translated doesn't have any stop codon. Okay, so thereby the ribosomes are stalled or stuck at the end of the RNA, mRNA. And third one, the no-go decay in this case is that because of the modification on the mRNA or the presence of a strong secondary structure, the ribosome cannot pass through these structures and the ribosome is stuck in the middle of the mRNA. So in the case of NMD in yeast, nonsense mediated decay in the case of yeast, what happens is that when the ribosome comes across a stop codon ahead of the natural stop codon, the stuck ribosomes or stalled ribosomes recruits protein factors, which in turn recruits exonucleases, which cleaves the poly A tail or de-adenylation of the mRNA is brought about. Okay, once it is de-adenylation happens, other exonucleolytic enzymes comes and degrade this mRNA. In the metazoans, the nonsense-mediated decay is slightly different from the yeast in the scenario that the stall ribosomes recruits release factors, which recruits other certain factors, which recruits endonucleolytic proteins, thereby cleaving the mRNA in the middle rather than deadenylation at the start. Okay, so metazoans non-mediated decay is slightly different. It brings about by the cleavage in the middle of the mRNA, and once it is cleaved, the mRNA is degraded by exonucleases. In the case of no stop decay, when the mRNA being translated doesn't have any stop codon, the ribosome goes all the way till the end of the RNA, even translating the poly A. What happens is that the ribosomes is stuck at the end of the mRNA, and these stuck ribosomes or stalled ribosomes recruits SKI factors, and SKI factors recruits exonucleases, which leads to the degradation of mRNA here. Okay, so this is the no stop decay. Now, no go decay is about when the ribosomes, there is no premature stop codon, it's not at the end of the mRNA, but it is stuck because of certain reasons like presence of strong secondary structure. Then, when the ribosomes stall, the DOM34 protein recruits other factors. The DOM34 is also involved in the NMD in yeast. This factor recruits endonuclease rather than deadenylases. It recruits endonucleases, cleaves the mRNA, just like the NMD system in metazoans. It recruits endonucleases and degrades the mRNA. So this is the no-go decay. Okay, so even in prokaryotes, I think I don't have the slide here, but I will include one definitely, is in the case of prokaryotes, this kind of stubs are there present in the prokaryotes as well. In the case of prokaryotes, when the ribosomes is stuck at mRNA, something like this, when there is no stop codon, what happens is that there is a RNA factor which is called the tmRNA or ssRNA that comes and rescues here, rescues the RNA and rescues the RNA by allowing translation to go further and terminating with a stop codon. In the case of prokaryotes, when ribosomes are stuck in the middle, either by the stop codon or by the presence of secondary structures, the system which comes in is that it recruits endonuclease and cleaves the RNA either at the stop codon or at the secondary structure site. It cleaves the mRNA, and the ssRNA or tmRNA that I just talked comes in and act the same way like the no stop decay in the case of prokaryotes. So tmRNA is very, very important for the prokaryotes to rescue ribosomes which are stuck because of secondary structure or because of the presence of stop codon in the middle, or the ribosome is stuck in the middle. Basically, what it does is that it generates a condition which is very similar to the ribosome stuck at the end of mRNA, and the ssRNA can come in and allow the translation to go ahead and terminate with stop codon. I will include a slide after this about the tmRNA, and with this, I would like to conclude the course, and in the following weeks, I will load some more questions. I will be sharing questions, and please go through those questions, try to understand the concept, because I will change the question little bit but remains the same concept, and it will have relevance in during the examination. Okay, so this one with this will be the last class, and let me know if you have any question at this stage. Anyone? Any question? Any question, guys?